The antimicrobial potential of the neem tree, part 2. Herpes and sulfonylglyceride, or Neem. Herpes simplex viruses, HSF, most commonly cause oral and genital infections. With an estimated 500 million people living with HSV type 2 and nearly 4 billion people with HSV type 1 in any given year. Because there is no cure for HSF infections and they can recur many times throughout a person's life, broad searches for novel antiviral medications are warranted in order to identify agents that may reduce the morbidity associated with these infections. These searches include phytochemicals isolated from medicinal plants such as neem. For example, the glycolipid cycloglyceride, SQDG, that has been isolated from neem leaves has been shown to have potent antiviral activity against HSV1 and 2, and half maximal effective concentrations were 9.1 and 8.5 respectively. The same study also found that HSF-infected SQDG-treated macrophages produce significantly less pro-inflammatory cytokines than untreated controls. It has been suggested that the antiviral and anti-inflammatory properties of SQDG may indicate that neem contains other phytochemicals with therapeutic potential against viruses. Concurrently, it has been found that 2 polysaccharides isolated from neem leaves, along with the sulfated derivatives, are able to inhibit HSV1 nucleic acid synthesis at concentrations that were not cytotoxic. Additionally, an aqueous neem bark extract was able to block HSV1 glycoprotein binding to it and virus entry into target cells in vitro. The preliminary data reported here indicated that products derived from neem can act as several steps of the viral life cycle to prevent herpes infections. Neem components against dengue proteins. Several groups have demonstrated that computational screening methods can be successfully used to identify novel inhibitors of viruses that infect hundreds of millions of people globally each year, including vector-borne disease such as dengue. In the context of dengue virus, 49 different bioflavonoids that are present in the neem tree were virtually screened for binding the DENV serine protease, NS2B, and S3 subsequent in vitro assays with promising candidates revealed that camphorol and epicatechin were able to inhibit the dengue infectivity by 77.7% and 62.6% respectively, without cell toxicity. Similarly, it was previously shown that 3 members of another important class of neem phytochemicals, the terpenoids, were able to bind with a high affinity in silico. This binding ability was subsequently confirmed in vitro. More of the common triterpenoids isolated from neem leaf extracts were shown to be effective against the envelope protein of all 4 types of dengue in silico. Overall, the ability of neem-derived phytochemicals to block the activities of both the protease and the envelope proteins of DENV and potentially other viruses further suggests that neem may be a source for antiviral drugs. Influenza and neem phytochemicals. Flu leads to an estimated 290 to 650,000 deaths annually. Due to these consistently high levels of associated morbidity and mortality around the world, influenza is one of the most intensely researched viruses. Though new vaccines for influenza are constantly in development, the burden of flu could be additionally reduced by the introduction of an antiviral that is effective at all stages of disease. Recent evidence suggests that neem may represent a robust source of novel drugs against viruses such as influenza. To summarize, molecular docking experiments identified a total of 4 Neem photochemicals that interact with conserved residues of either the nucleoprotein or the non-structural protein of influenza. Though requiring further testing, this may indicate the ability to act as a universal drug against the flu virus. Neem and SARS-CoV-2. Taking the lives of more than 5 million people over the last 2 years. SARS-CoV-2 and the associated disease COVID-19 continues to be a significant threat to public health for which there are few treatment options. Consequently, many researchers have successfully screened chemical libraries for viral inhibitors that act against SARS-CoV-2. Detailed reviews on the potential for medicinal plants to be used against SARS-CoV-2 are available. Phytochemical and plant-derived products like those from Neem are sometimes included in these chemical libraries. For example, Neem binding simulations identified nemomolecular as a compound with strong affinity for both the main protease and the spike protein of SARS-CoV-2. Similar docking studies found that nemomolecular binds to the papain-like protease of SARS-CoV-2 with higher affinity than remdesivir, indicating its potential to hinder viral replication. Although these types of studies are high throughout and can detect candidates for further investigation, the virtual studies must typically be followed by in vitro experiments before an antiviral can have clinical relevance. Despite this, given the devastation associated with the ongoing pandemic, several clinical trials are in progress to test Neem as a component of mouthwashes, nasal sprays, and capsules for suspected, confirmed, and hospitalized COVID-19 patients or for prophylaxis. Additionally, one animal study has been completed in which coronavirus-infected mice were treated orally or intranasally with neem bark extract. Overall, the therapy was able to prevent systemic injury and pathologic effects of the virus, both in vivo and in vitro, using multiple model systems. Advocating for additional trials of Neem against SARS-CoV-2 made connections between some of the known mechanisms of Neem products against other illnesses and the potential efficacy of Neem against the virus that causes COVID-19. For additional information about the pharmacological implications for the use of medicinal plants against respiratory infections, see the recent review. All in all, the antiviral action of Neem against a variety of viruses can cause human disease is evident. Moreover, outside of HIV, HSV, DENV, influenza, and SARS-CoV-2, there is a strong indication that the aforementioned phytochemical screening methods may be used to identify novel inhibitors of other viral life cycles. Antiparasitic evidence. Although the area of research regarding Neem as a potential therapeutic agent against parasites is relatively underdeveloped, most of the investigations that have been published so far have focused on Plasmodium, the human pathogen of global importance that causes malaria. More than 500,000 deaths attributed to malaria are estimated to occur each year. While there are effective prophylactics and therapeutics approved for this disease, these resources are not readily available in all parts of the world. In these instances, there is the possibility that natural products may be more easily acquired, distributed, and accepted. Neem products could be a part of the solution. Recently, a comprehensive review on the antiplasmodial activities of African medicinal plants was published and found that Neem extracts consistently performed well across experiments. Studies using both in vitro and in vivo models indicate that aqueous, methanolic, and ethanolic extracts of Neem stems, leaves, and bark all have significant anti-malarial activity against a variety of Plasmodium and Plasmodium becket strains. The publications suggest that neem and other medicinal plants are active against malaria. However, one clinical trial with the African plant species has been conducted so far, and despite the lack of controlled human studies, the malaria mouse model has been used to demonstrate that neem bark and seed extracts are active against Plasmodium infection, both alone and as part of the polyherbal mixture in the case of the bark extract. This was indicated by parasite suppression of decreased erythrocyte infection. The gestational malarial mouse model has also been shown that administration of neem leaves improves the overall health of the infected dams, including reduced parasitemia, increased platelet counts, lower levels of preeclampsia biomarkers, and increased birth weight of pups. Additionally, the limonoid, which was found within neem seed extracts, is able to interfere with the early sporogony stages. This finding suggests that certain neem-associated phytochemicals may have the ability to limit Plasmodium transmissibility, warranting further investigation in other models and clinical trials. Another commonly studied phytochemical of neem, azadirachtin, was shown to bind to the Gefrin E almost as well as artesunate during its in silico analysis, which indicates that upon further study that neem may be effective treatment for cerebral malaria. Providing additional convincing data for the investigation of neem products as therapeutics used in the mouse model, the infection to show that an aqueous neem leaf extract was able to reduce the blood markers of malaria-induced renal injury to normal levels without being toxic to the animals. Taken together, the available in vitro and in vivo data suggests that plants used in traditional medicine may be further explored in clinical trials for their anti-malarial capacity. Throughout tropical and subtropical areas around the world, the neglected tropical proteacean Leishmania causes an estimated 700 to 1 million infections each year, and is highly fatal if left untreated. Visceral leishmaniasis is the most severe form of associated disease. The leishmaniasis can be treated, but there is not currently a drug available that eliminates the parasite from the body. Thus, the patient is susceptible to relapse if immunosuppression occurs. Although studies on this topic are limited, there is some evidence that plant extracts can kill the parasites, including an ethyl acetate fraction of neem leaf extracts. When treated with this, that underwent an apoptosis-like death, and intracellular ambitotis was also killed in vitro and in vivo. With this evidence, more in vivo and translational research on the activity of neem is merited. Finally, the last antimicrobial activity of neem that has been recently explored is that against the parasitic worms. As the most widespread neglected tropical diseases globally, the burden of schistosomiasis and soil-transmitted infections could undoubtedly be reduced with the addition of new and accessible plant-derived treatments or prophylactics to already available drug regimens. Although still a minor area of research, the anti-schmutz and antithalamic properties of neem suggest that natural products can be potent inhibitors of larger eukaryotic pathogens as well. Indeed, the same neem leaf extract that was effective against MRSA biofilms also caused severe morphology changes, significant reduction of motor activity, and the death of the worms in vitro. For helminth-related studies, in vivo experiments showed that neem leaf powder used at 500 milligrams per kilogram body weight worked as well as 5 milligrams per kilogram of fensbenzodiazole to treat cows with bovine Trongleus is a major hurdle to profitable farming in tropical and subtropical regions and can have a substantial economic impact. Similarly problematic things for the poultry industry, which are parasitic tapeworms. These organisms were found to be severely paralyzed, damaged, and killed by short exposures to the glyptolipid form of neem extracts that also has antiviral activity. Although not yet tested on humans, the neem-derived products appear to have strong potential against animal pathogens, thus supporting the need for a deeper investigation of the overall anti-parasitic activity of neem. The future of neem as an antimicrobial is summarized in this review. A variety of extracts and phytochemicals from the Indian evergreen tree A. indica have significant antimicrobial activity against a multitude of pathogens that affect human health. The dental applications of neem appear to be one of the most researched areas, with the general antibacterial properties of neem not far behind. Intriguingly, there are several recent studies that suggest that neem may have broad applications in the food industry, aside from its traditional uses in agriculture, for pest control and fertilizer. Some of the least developed areas, however, are the antiviral and antiparasitic activities of neem-derived products. Overall, there is significant evidence to warrant further investigation into these properties of neem. Undoubtedly, the models will be crucial to understanding the clinical relevance of neem against all of the microorganisms mentioned here and those that have yet to be tested. Of note, in recent years, there are several groups who have incorporated neem into novel materials and technologies that have broad implications for human health, specifically green synthesized copper or silver nanoparticles and hydrogels, nanocellulose films, chitosan copper oxide biopolymers, and hydroxyapatite have all been constructed to include neem extracts and have substantial antimicrobial activity, including against multiple drug-resistant bacterial species. Both in vitro and in vivo data suggest that these composite materials represent a growing industry of creative antimicrobial technologies that have the potential to revolutionize infectious disease treatment and biomedical science as a whole. In addition to the many recently published manuscripts that indicate that neem-derived substances have broad-spectrum antimicrobial properties, it is worth noting that dozens of patents are filed each year that mention neem-based products. Indeed, a similar query of the United States Patent and Trademarks Office online patent public search tool using the terms neem and antimicrobial yields over 400 results since 2015. This available list of patents helps to demonstrate the limitless applications of the antimicrobial properties of neem. While too many exist to cover in detail, some notable examples include very diverse applications. For example, medical gloves and a polymeric yarn are for use in hygienic textiles, have both been enriched with neem derivatives and shown to have beneficial antimicrobial properties. Furthermore, in line with the reported antimicrobial uses of neem in the fields of dentistry, dermatology, and agriculture, many of the patented neem-containing products fall into these categories as well. Patents have been awarded for neem-containing dental rinses and composites, for neem and for other plant extract containing topical treatments for mild skin disorders, and for neem-based pest control formulas. Clearly, the investigation of neem as an antimicrobial is an area of research that is constantly expanding and is generating valuable products that may improve human health. In order to develop realistic neem treatment regimens that could be used in humans, there are clearly many intriguing areas for future investigation. Undoubtedly, future experiments will need to elucidate the mechanisms of action of neem and the associated phytochemicals. Given the available data summarized in this review, Some of the most promising areas of investigation moving forward appear to be the application of individual neem phytochemicals and derivatives thereof as antimicrobial agents alone and used in combination with existing treatments. Additional preclinical and clinical studies to determine the toxicity and effect of in vivo dosing of specific phytochemicals as compared to parent neem extracts, and the inclusion of hundreds of available medicinal plant products, extracts, and phytochemicals in screens for potential inhibitors of emerging and resistant infectious diseases. It's important to note that to gain maximal utility from these areas of research, close attention should be paid to the types of extracts, including both the particular part of the plant and the solvent, that have already been tested against which organisms. Some level of standardization should be considered so that comparisons can be made and patterns can be recognized across multiple studies. This may become easier when the antimicrobial activities of more individual phytochemicals are determined in mass. Neem represents a novel source of antimicrobials that may be used to combat drug resistance and emerging threats to human health. Furthermore, the research that has been done on the neem tree can be used as a guide to encourage the investigation of other traditionally used natural products for their utility as modern pharmaceuticals. The research in the Merrill Lab is supported by funding from the NIH and the United States DoD.